going to walk you through the steps I can. Okay. So we're going to start with the pathogen. What is a pathogen?
Disease-causing organism.
Germ, invader, disease. Pick a word that you want to work with. For tonight, we're going to use the word non-self to represent the pathogen, because pathogens are not me, right? So the non-self entity enters a body cell. How did it get into a body cell? I wonder.
Phagocytosis.
Very good. So yeah, it goes through my barriers, goes through all my skin and whatnot, and starts to enter my cells, which are self, right? Then this is going to happen. The cell is going to take the invader and try to present the antigen. So I'm going to show you a different view of this. Okay? A little different picture. And I'll show you the animation. Right here. Pleasure. Uh-oh. Big immunity, right? On this picture, non-self over there. Non-self has entered a body cell. That's me, because here's the MHC. And what is my body cell doing?
Processing it.
It's processing it to do what? Where's it going to stick the non-self antigen?
On the MHC protein.
On an MHC. So eventually I'm going to stick the viral non-self antigen on the tip of my antigens. Right? So we call this the presentation stage. So in step number 2, the body cell presents the non-self antigen. It sticks the non-me part on the outside. It announces. So if you do that, if you are a cell that presents non-self antigen, we call you an antigen presenting cell. Anyone know the abbreviation for an antigen presenting cell?
APC.
An APC, right? An APC in this context means a cell that's going to present the antigen. Right? I'm going to present it. Where am I presenting it? On the end of the what? On the end of on the self cell. So the non-self on end of the self. That's the antigen presenting step. We used to call this the trophy step, right? Look what I found, right? I'm presenting that. So now we got to figure out types of cells that might do this. So there are certain cells that do this more than others. Can anyone guess over here what cells might be encountering lots of non-self things?
White blood cells.
Yeah, the white blood cells are the ones that are most likely to run into a non-self entity, right? Especially number 3. The monocytes are most likely these ones. So other cells can be APCs, but the monocytes are the most likely ones because they eat things. So it's very likely that they would be the ones that would then present themselves to the body. So in this picture, I have a body cell, which could be a monocyte, presenting the antigen. Let me show you an animation of that process. Right here. Lost my picture. There we go.
Certain phagocytic cells of the immune system, called antigen-presenting cells, seek out foreign cells and engulf them. In addition, despite the best efforts of the immune system, cells can become infected. When this happens, the immune system needs a way to see inside the cell in order to know that it is infected or has found invaders.
Bingo. That's the most important concept is if I'm an invader and I get inside of you, you have no way of knowing where I am. So these cells are then going to present you to the outside world so you can see that you're infected. I have to warn the body that you're there. That's the concept of an APC.
Mammalian cells use antigen processing and presentation to accomplish that task.
Indeed we do. Slide 2, slide. All right, so let's do that. Let's walk through the APC stuff. All right, so let's do that.
Some of the protein molecules synthesized within most nucleated cells are digested into smaller peptides, about 8 to 10 amino acids in length. These peptides enter the endoplasmic reticulum and are loaded onto MHC proteins, which are products of a group of genes called the major histocompatibility complex. The vesicles containing the MHC peptide complexes bud off of the endoplasmic reticulum and are released into the cytosol.
That's all 112 stuff, right? So these are proteins. So they're made by the ER, transported by the Golgi to cell surface.
Vesicles containing the MHC peptide complexes travel through the cytosol to the membrane. The vesicles fuse with the membrane, delivering the complexes to the exterior of the cell.
We also call these markers tags. Right?
MHC proteins are used to present peptides on the surface of the cell. Since the peptides are produced from proteins inside the cell, they allow cells of the immune system to detect proteins inside the cell without entering the cell.
Also called wearing your heart on your sleeve kind of logic. So I have to show the outside of what's inside of me.
Special antigen-presenting cells, including monocytes, macrophages, and their relatives, engulf invading pathogens and digest them, producing fragments with antigenic determinants.
So I'm eating the invader, I'm a white blood cell. Now I got to announce to the world.
Vesicles containing MHC proteins fuse with vesicles containing digested pathogens. The antigenic determinants from the pathogens are loaded onto the MHC proteins. MHC proteins are used to present antigenic determinants on the surface of the cell. The association of the antigenic determinants with specific MHC proteins signals immune cells that these antigenic determinants came from digested invaders.
Okay. So what I'm doing, again, is I'm taking what's invading me and then putting it on the surface. So why? Who am I presenting it to? I'm presenting it. Which other cell in particular? Let's look at this picture. Can anyone see what cell is being presented to?
The helper T cell.
The helper T cell. Step number 3. Where? Step number 3. So the APC presents to the helper T cell. So that's the cell I'm trying to get the attention of, is the helper T cell. Okay, so just for your viewing pleasure, you can abbreviate helper T cell as a T with an H. So if you want to save yourself some time, that's the abbreviation for helper T cell. Or you can just write helper T cell. So this thing here is a helper T cell. It's noticing, quote, quote, that thing there. So I'm presenting to my helper T cell. Whoop-de-doo. What's a helper T cell going to do?
Spread the message.
It will. So a helper T cell activates other immune cells. So a helper T cell is going to activate, turn on, stimulate, pick a word you like, other immune cells. Okay. So, what other immune cells are those?
Neutrophil.
Very good. Those immune cells are other T cells, or, can you guess what the next one is?
B cells.
B cells. Okay, so no matter which B or T system I'm talking about, we're going to do the same first 5 steps. I have to have an APC telling a helper T cell what happened. After step 4, basically step 5, then you choose 2 different pathways. All right? But first what we're going to do is go back to the animation and show you what we've got so far from step 1 to step 5. All right? So we're going to redo this one more time. The different, not that one. This one here.
Activation of the immune response typically begins when a pathogen enters the body. Macrophages that encounter the pathogen ingest, process, and display the antigen fragments on their cell surfaces. Macrophages with antigen fragments displayed on their surfaces are called antigen-presenting
Or
cells.
an APC.
An antigen-presenting macrophage interacts with a T helper cell that can recognize the same antigen.
Boom. Okay. So that's basically step 4. Right? There's a magic word in that narration, in that last sentence. Can we see the magic word?
Interacts.
Besides interact, that's a good word. I'm looking for.
Recognizes.
Let's see, blah, 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 blah. This one.
Same.
Same. Doesn't seem very magical, but what does that mean, same antigen? Would that be non-specific or specific?
Specific.
Is that concept of specific? I'm not going to attack everything. I'm just going to learn this shape here. That's the only antigen that I am presenting. Nothing else. So your innate defenses will kill anything. These are only going to do something with that shape there. No other shape. So this is where that specific nature comes from. I'm only going to do something to that. Right? So I'm going to get the specific concept down. So it isn't every antigen, it's just that one. Okay?
During the interaction, the macrophage releases a chemical alarm signal called interleukin-1, which stimulates the T helper cell to secrete interleukin-2. Interleukin-2 causes the proliferation of certain cytotoxic T cells and B cells.
Okay, so that's where we're at with step 5. So now we're going to turn on B's and T's. But we need to write down that chemical because P says thou shalt know it. What was the chemical that the helper T cell used to turn on the other cells?
Interleukin.
Interleukin-2. That's a chemical, that's essentially a hormone. Chemical messenger. That activates the B's and the T's. Interleukin-2. I broke it up in 2 words so you get the idea of what the word means. What does inter mean?
Between white
leukin,
white cells.
white cells. So it's between the white blood cells. That's the logic of the name. So the helper T cell is going to give a chemical order to the B's and the T's. So in the Army, what do you call the guy that gives all the orders?
Officer.
Officer or general. So the helper T cell is the general, the admiral, the officer, the boss. Pick a word you want to use for your analogy. But the helper T cell is giving orders to the rest of the immune system. It orders them around. It won't do anything, that's what officers do. They just give orders. Right? This is the logic here. So the T and B cells are going to do the work. The helper T cell gives the orders, gives the instructions. Make sense? So the helper T cell is the general, the officer, the commander, the admiral, the captain, the teacher. Make sense? All right, so far we've turned on our helper T to turn on our B's and T's. Now we're going to have 2 sets of nodes. You're going to have a T cell set of nodes, and you have a B cell set of nodes. So I would split your paper for me, but I can't put them all on the board at once. So we're going to do 2 sets of nodes, B's and T's. We're going to start with the T cells, and then we'll go back and do B cells.